All right. Okay. So in this second part of the lesson, we are going to talk about neurulation. So the formation of neurulation is the formation of the neural tube. So which tissue, which layer of cells are responsible for the formation of the neural tube? Well, it is the ectoderm. The ectoderm is responsible for the formation of the neural tube amongst other things. Other things. In fact, the ectoderm is responsible for the formation of the surface epidermis, so the surface ectoderm or epidermis, and the formation of hairs and nails, for instance. And the cells of the ectoderm that will become epidermis express high level of BMP. However, the cells of the ectoderm that are going to become the neural tube, and the neural tube will give us the brain, the spinal cord, and the motor neurons, for instance, as well as the retina, express low level of BMP. So depending on the level of expression of BMP, the cells will go in one direction and, for example, become surface ectoderm, or another direction and become a neural tube. Cells that express intermediate level of BMP are the ones that become the neural crest. And the neural crest are responsible for the formation of the peripheral nervous system, for instance. So depending on the level of expression of BMP, the ectodermal cells will take different jobs and they become different things. Either surface ectoderm, therefore epidermis, neural crest, or neural tube. The embryo, when is undergoing neurulation, is called a neurula. So a neurula is the embryo undergoing neurulation. Here we have a summary, if you want, a very simple summary of an amphibian neurulation. Remember, amphibians are vertebrate, so and their development is very similar to mammals, which is very similar to humans as we are mammals indeed. On the top, three pictures, the bluish pictures, we are observing neurulation from the top, while on the bottom three pictures, we are observing neurulation from a section of the animal, toward a bilateral section, and we are looking toward the trunk of the animal, of the embryo. So what we can recognize in the first three pictures is that at one point the ectoderm form some thickening, which is called a neural plate, and this neural plate are pushed toward each other. So this neural plate is pushed toward each other and form a primordial neural tube. And the two parts of the neural plate are fusing in the middle, forming a neural tube at the end of neurulation. If we look toward the section, we can recognize the neural plate, which is a thickening of the ectoderm, forming above the notochord. In fact, it is the notochord that induces by signaling the formation of the neural plate in the first place. Then the neural plate, the two sides of the neural plate are pushed toward each other, forming a groove, and then this groove is closed by the neural plate tips fusing to each other and forming a neural tube. Notice how the neural tube is actually formed on top of the notochord. Now, the neural tube formation can happen by primary or secondary neurulation. Now, primary neurulation and secondary neurulation occurs both in the same animal. Okay, so it's not that some animals have primary neurulation, some other animals have secondary neurulation. Granted, there are some species that only have primary neurulation, but in all mammals and in most vertebrates, in the same embryo, we have both primary and secondary neurulation. Primary neurulation happen in the anterior part of the embryo, while secondary neurulation happen in the posterior part of the embryo. Okay, so in the anterior part of the embryo, we have primary neurulation. In the posterior part of the embryo, we have secondary neurulation. In primary neurulation, we can see it here in this left-hand side of this picture, the ectodermal cells, so the external ectodermal cell, form a thickening, which is the neural plate. The neural plate sides are pushed toward each other, and then the tips of the neural plate fuse to each other and detach from the rest of the ectoderm that is going to form the surface epidermis. Notice how here the cells that are responsible for the formation of the neural tube are initially attached to the cells that are responsible for the formation of the epidermis, and they need at one point to be resolved from each other. On the other hand, in secondary neurulation, which happen in the posterior part of the body, this doesn't happen. What does it mean? It means that a group of mesenchymal cells that are both ectodermal and mesodermal in origin form a medullary cord above the notochord. And this medullary cord then form cavitation, so they form a lumen, and this lumen then become the neural tube. So what's happened here, and is important to remember, is that the cells that are forming the medullary cord and therefore become the neural tube are never in contact with the external ectodermal cell that are going to form the epidermis. So why do we have primary and secondary neurulation? If we look at different species, here we have the spine structure of a typical bird, and here we have a spine structure of humans, and here we have an amphioxo, so which is one of the a vertebrate, a primordial vertebrate, if you want. So we find primary neurulation usually connected to the neural tube that is responsible for the formation of the anterior part of the spine and the brain. In fact, the anterior part of the neural tube will develop into the brain. Okay, and the posterior part of the neural tube will develop into the spine. So primary neurulation is usually associated with this part of the body. In some species, like amphioxus, we have only primary neurulation. So the entire spine is produced by primary neurulation. However, in most mammals and most vertebrates, secondary neurulation is associated with the posterior part of the spine, and it is probably associated with, in some species, the formation and elongation of the tail. We also recognize a transition zone between primary and secondary neurulation. In this transition zone, we have a neurulation, and I show you now, neurulation that has characteristic of both primary and secondary neurulation. This is called the junctional zone or junctional neurulation. In this situation, we have that both a thickening of the external ectoderm is present and forming a primordial neural plate, as well as a mesenchymal cells organization is recognized. So both the neural tube is both formed by these mesenchymal cells, like secondary neurulation, and the neural plate pushing and detaching from the ectodermal cell that are responsible for the formation of the epidermis. Now, one characteristic of primary neurulation, or neurulation in general, is that it goes it forms the neural tube from an anterior to posterior position. So if you remember, and it's important that you remember this, when we were talking about the formation of the node, we said that the node is formed posteriorly and then it moves anteriorly. Then when you reach the most anterior position, the node started to regress posteriorly. While it is regressing posteriorly, the notochord is formed, and the notochord therefore is formed from anterior to posterior position, following the regression of the node. Now, because it is the notochord that induced the formation of the neural tube, while the notochord is formed, and therefore from anterior to posterior, it induced the above cells to become the neural tube. And therefore, the neural plate initially, which will become the neural tube, is formed in an anterior position and then regress posteriorly, okay, following the formation of the notochord. So what's happened is that here we have a nice picture, a nice cartoon that represents different part of a chicken embryo during neurulation. And what we see in the same embryo at the same time, we have on the anterior part of the embryo, where the neural tube is almost completed and almost closed. So you see here the neural plate that are almost forming the neural tube above the notochord, which is always there, while more posteriorly, the neural plate are just starting forming their groove, and even more posteriorly, we recognize still the primitive streak, so we don't even have started neurulation. So while more anteriorly, neurulation is almost complete, more posteriorly, neurulation still has to happen. And actually, the embryo is finishing gastrulation. So what we have here, on top of it, and I already mentioned, is that the anterior part of the neural tube, when it is formed, is going to develop into the brain, while the posterior part of the neural tube is going to develop into the spine. So here we recognize the notochord. The notochord, of course, is always inducing the formation of the neural tube, so it is below. We find it below the neural tube. Here we have the neural folds, which are basically the folds of the neural plate. And here we can recognize the thickening of the neural fold, so the initial neural plate and the thickening of the neural folds. So let's have a look more closely at what's happened during primary neurulation. So initially, cells that are induced by the notochord start to divide, and they divide mostly in the anterior-posterior direction or in the anterior-posterior direction. So they also contribute to elongate the embryo itself. Then we have the formation of what is called the medial hinge point. So the medial hinge points are formed just above the notochord. The notochord induced the formation of this middle hinge point. Basically, it induced a change in shape of the cells above the notochord. And because the cells change shape, they push the two sides of the neural plate toward each other. They change shape, and so they push the two sides toward each other. In doing so, we have the formation of the neural groove. So the neural groove is visible when the hinge point is formed. Okay? So if Taffy9 would happen, the two sides will come together, forming a bilayer. But what is happening actually is that we have the formation of two extra dorsolateral hinge points, which bend the two tips of the neural fold or the neural plate toward each other. So we have the first one, and then the second ones. They push the two tips toward each other, forming actually tube. So the two tips, when they are in contact with each other, they fuse together, they detach from the ectoderm that is going to form the epidermis, and they when they fuse together, they form the actual neural tube. Some of the cells here, in between the neural tube and the external epidermis, are released, and these released cells are the neural crest cells, and they're going to form the peripheral nervous system. Okay, but how do the cells in the neural plate know that they need to become hinge point? Well, there is a number of signals that start from the notochord that give instruction to the cells of the neural plate to become hinge point or not. For instance, the cells just above the notochord receive SHH signals from the notochord itself. So the notochord express a lot of SHH, will induce the cells just above it to change shape and therefore become hinge point. So noggin is also expressed in the neural. Noggin is expressed in the neural folds, in the tips of the neural plate, and induce the formation of the dorsolateral hinge points as well. So noggin is important for the formation of the DLHP. But the expression of noggin is antagonized by SHH. So this is important because therefore not all the cells of the neural plate are becoming hinge point. Only a group of cells that receive enough noggin signals and but not too much because of SHH is blocking it. So the cells that are just far away from the point of expression of noggin, which is the neural, the tips of the neural plate, cannot become DLHP. On the other hand, noggin inhibits the expression of BMP. BMP is highly expressed in the ectodermal cells that are going to become epidermis. And BMP is an inhibitor of both DLHP and MHP. So it is an interplay between BMP, noggin, and SHH that control the formation of both the medial and lateral hinge points. Okay. So let's now talk about secondary neurulation. So we talk about primary neurulation, but let's talk about secondary neurulation.